Ever catch yourself thinking about how we figure out left from right? It seems so basic, but when you realize it happens before we even have a brain, it's kind of mind-blowing, right?
Definitely.
And that's what we're getting into today, how our bodies do that whole left-right thing way back during, get this, embryonic development.
Always a fascinating topic.
We're diving deep into a scientific article. Ready for the title? Asymmetries in HK, ATPases, and cell membrane potentials comprise a very early step in left-right patterning.
Catchy.
I know, right? But it's got some wild stuff in there. And we've got our expert here to help us break it all down.
Happy to be here. Always up for a good deep dive.
So we all know our left and right sides aren't perfectly symmetrical, but it goes way deeper than that. Our organs are in the mix too.
Exactly, and that's not just by chance. That left-right setup is crucial. Heart, liver, lungs, it all depends on that early process. When it gets messed up, like in situs inversus where organs are flipped, it can cause problems.
And sometimes it's even, well, you know.
Fatal, yeah. Completely symmetrical development just doesn't work.
Right. So how does it all go down? How does a tiny clump of cells, an embryo, figure out left from right? That's what this research is all about. They're looking at some surprising subjects, frog and chicken embryos.
And the really cool part is they're focused on the very first steps, way earlier than we used to think. It's like peeking at the very first page of a mystery.
Okay, let's hear about these frog embryos. What did they actually do?
So they used African clawed frog embryos, Xenopus, pretty standard in developmental biology. They were tracking this molecule, HK, ATPase, don't get too hung up on the name, it's basically a pump, moving these charged particles called ions across cell membranes, crucial for a bunch of cellular stuff.
It's a tiny pump, big role.
Exactly. And here's the kicker. They found that the instructions to make this pump, the mRNA, it clusters on one side of the embryo super early. We're talking within the first 2 hours after fertilization.
2 hours, that's practically nothing. It's wild that such a big difference is already there at that stage.
Totally. And it gets wilder. This early asymmetry with the HK-AT pace seems to be connected to electrical differences within the embryo.
Wait, electrical differences, like the embryo is running on electricity.
Not quite wires and circuits, but you're getting the idea. Those ion pumps, they don't just move ions randomly. They create differences in electrical charge, like a teeny tiny battery.
So like a microscopic electrical language.
Exactly. And this research is saying this electrical chat is key for setting up that left-right difference in the embryo.
So from frog eggs to microscopic electrical currents, what's next? How do we get from those signals to a whole organism with everything in the right place?
That's where the chickens come in.
Okay, hold on. Before we move on to the chickens, let's take a quick break. All right, so we were in the thick of it before the break. Frog embryos, microscopic currents, the works. That whole idea of tiny signals being so important still has me kind of floored.
It really is something else, and it makes you realize how much we're still figuring out about, like, the very beginning of life. We're talking mechanisms that have been around since forever, and we're just scratching the surface.
It really is a whole other world at that scale, huh?
Absolutely, and it's like it just keeps getting more amazing.
Okay, so we left off with those electrical gradients in the frog embryos. What happens next in the research? Where do the chickens come in?
So they got these great results with the frog embryos, and they wanted to see if they could actually see these electrical patterns in a developing embryo. For that, they turned to chicken embryos, another big player in developmental biology.
Right, chicken embryos. But how do you even measure electrical signals in something that small? That just sounds, I don't know, impossibly difficult.
Oh, it is. But these researchers, they use this really clever technique where they could pick up even the tiniest differences in electrical charge. It's a bit technical, but basically they use this special dye that changes color depending on the electrical potential of the cells around it.
So they could actually see the patterns, like with their eyes.
Kind of. They got these amazing pictures of the developing chicken embryo, and you could clearly see different electrical areas on either side of its midline, like a dividing line between left and right.
Wow, so it wasn't just theory, they actually saw these electrical gradients. That's incredible. But how would they know it was connected to the H/K ATPase, the pumps we were talking about before?
Good question. They did some really smart experiments to test that link. Turns out, if they messed with how the HIAT-PASE worked, poof, the electrical differences vanished. And get this, when they manipulated the pumps to create artificial gradients, it actually changed how the embryo developed.
So they were controlling the electrical signals with these tiny pumps, And that changed how the embryo grew. That's huge.
Exactly. And that's why this research is so groundbreaking. It's saying these gradients aren't just a side effect, they're actively shaping how the body becomes asymmetrical.
It's like they're rewriting the rules of embryonic development.
In a way, they are, and it goes way beyond chickens and frogs. This has got people thinking about electrical signaling in human development and even diseases.
Right, because if it's this crucial in chickens and frogs, who's to say it's not the same in us?
Exactly. And if it is, it could change how we understand birth defects and maybe even lead to new treatments.
Now that's what I call a deep dive.
Right.
We've gone from frog ponds to chicken embryos to the future of medicine.
And we're not done yet. There's one more part of this research that blew my mind. It turns our whole idea of left and right on its head.
Okay, now you've really got my attention. What do you mean?
Well, we tend to think of our left and right sides as mirror images, right? But when it comes to how we develop, that might not be the whole story. What if, and this is where it gets really interesting, what we call our left side is actually the default setting for an embryo?
Hold on, are you saying everything we think we know about left and right could be backward?
In a way, yeah, it's like a totally new way of thinking about our own bodies.
Okay, this is getting pretty deep. So if our left side is the default, what does that make our right side?
That's the million-dollar question. It implies our right side is actively patterned, like it's responding to signals from the left.
So instead of a mirror image, our right side is more like a carefully planned response to the left.
Exactly. And that opens up a whole new world. What are those signals? How do they do that? And what happens if they go wrong?
This is mind-blowing. Our understanding of left and right just got flipped.
It really makes you think, doesn't it? And it just shows how much we still don't know about biology.
Totally. This research has been quite a ride, and I'm so glad we could share it with everyone today.
The pleasure is all mine. Always great to talk about science that's pushing the boundaries.
Right. Before we wrap up, though, there's one more thing. We talk about birth defects, but what about regenerative medicine? If we can understand these electrical signals, could we use them to, I don't know, regrow damaged tissues or even organs someday?
Now that is a fascinating thought, and you know what? Researchers are already on it. This whole field of bioelectricity, looking at how electrical stuff affects living things, it's still early days, but it's got huge potential for regenerative medicine. Imagine using electrical stimulation to, help regrow heart tissue after a heart attack, or even repair spinal cord injuries.
That sounds like science fiction.
Right, but the more we understand about electricity and biology, the closer we get to making that science fiction a reality.
This has been an incredible conversation, and thank you so much for joining us today and sharing all this incredible stuff.
Anytime. I'm always happy to geek out about cutting-edge science.
And for our listeners, we'll link to the research we talked about in the show notes so you can check it out yourself. But for now, we're going to take a quick break. Hang tight. Okay, so we've covered a lot today.
We really have.
From frog ponds to chicken embryos to, the whole idea of left and right being completely backwards.
That's a lot to take in.
It really makes you think about how much we take for granted, you know?
Yeah.
Like left and right, so simple, but it turns out it's incredibly complex.
Absolutely.
It really makes you appreciate what science can do, challenging what we think we know and uncovering these incredible things about how the world works, even just inside our own bodies.
It's true, and we've only just scratched the surface with embryonic development, haven't we? Every time we think we've got a handle on things, boom, more questions pop up.
Exactly. And speaking of questions, this research, it leaves us with a lot to think about.
For sure.
What does this all mean for, evolution? If this electrical signaling is so important for development, does it go all the way back to the very first animals? How did this whole intricate system even come about in the first place?
Those are the million-dollar questions, and you know what? Scientists are trying to figure that out right now.
Yeah.
Some people think bioelectricity, that whole electricity meets living things thing, it might have been a major player way back when life was just getting started.
Wow,
It's a huge area of research, and it could unlock some major secrets about, where we come from.
It's kind of mind-blowing when you think about it, that something as, fundamental as electricity could be so deeply connected to life itself.
It really is.
Well, I think we've done a pretty good job of, breaking down this whole complex topic today.
Definitely.
But before we go, I want to leave everyone with something to, maybe chew on for a bit.
Okay, I like it.
We've been talking about left and right in a very physical way, but what about a more metaphorical way? You know how we talk about left-brained versus right-brained thinking, or left-wing versus right-wing politics.
All right.
Do you think those early developmental things we talked about, those electrical signals shaping our physical left and right,
Mhm.
do you think those could have any connection to these bigger, more abstract ideas?
That's an interesting thought.
It might sound a little out there, but I think it's worth thinking about. If our whole understanding of left and right needs a rethink in biology, maybe it's time to re-examine those ideas in other parts of our lives, too.
I like that.
So, to everyone listening, we'll leave you with that thought. We hope this deep dive got you thinking and maybe even gave you a new appreciation for the, I don't know, the just amazing world of embryonic development.
It is pretty amazing.
And who knows, maybe it'll even make you look at the world, or even yourself, a little differently.
You never know.
Thanks for joining us, everyone, and until next time, keep exploring.